Good evening. So before we get started, I forgot to mention something to the first two labs, the 11 o'clock and the 4:30 lab. On Wednesday, we are starting our disinfectant lab, and this will allow us to test our unknown against different cleaners, different disinfectants and things. And so we have a standard set that we use. But I'd like to encourage you, if you have anything at home that you would like to test on your unknown, bring a little sample of it in, and we'll be able to test its effectiveness against your microbes. So if you have a dishwasher, dishwashing liquid, or some other kind of hand sanitizer or cleaner that you want to see how effective it really is, go ahead and bring it in, and we'll test it on your unknown. So a couple of current events tonight. Current event number 7 is actually two different articles, but they both deal with the same subject matter, recall of food because of a salmonella infection. So let me read the first one to you, a little short one. Dole Foods, Monterey-based fresh vegetable division, is recalling 756 cases of bagged salads because they could be contaminated with salmonella. The bags of 7 lettuces salad were distributed in Alabama, Florida, Illinois, Indiana, Maryland, Massachusetts, yada, yada, yada. The company said the bags are being recalled because a random sample tested by the state of New York came back positive for salmonella. No other Dole salads are included in the recall. And then 2 days prior to that, another salmonella recall. This one, a Cupertino firm, pulls back tuna product that left 116 people ill. A Cupertino-based company is recalling a tuna product after it was linked to a salmonella outbreak that sickened 116 people around the country, though none in California. Moon Marine USA Corporation, or MMI, voluntarily recalled over 58,000 pounds of frozen raw yellowfish product labeled Nakaochi. Scrape AA and AAA, the Food and Drug Administration reported. The food is scraped off bones and looks like a ground product. It is not for sale individually, but is used to make sushi and similar dishes. So far, 12 people have been hospitalized, but nobody has died. That's good news. So salmonella, you hear about it often, especially in cases like this. You know, what is it really? It's a microbe, it's an intestinal microbe that is gram-negative, very similar in a lot of respects to E. coli and to Shigella. And in fact, as we gain more and more DNA evidence, there is some that are suggesting Salmonella, Shigella, and E. coli are all related, with differences developing over time, of course. Salmonella, you get it through ingestion. Usually you need a large number of cells because it doesn't survive well in the stomach where there's a high acid concentration. However, once it gets into the small intestines and the large intestines as well, it can adhere to epithelial cells. And then we talked about the type 3 secretion system. It uses this type 3 secretion system to induce the cell to bring the microbe inside. So now it's in a phagosome. Okay, it's been engulfed and it's in a phagosome. The microbe multiplies in the phagosome, so it doesn't get digested in the cell, and eventually it gets distributed on the base side of the cell. We talked about the sampling method that our epithelial cells use. Generally, by the time it gets there, our macrophages are able to eat most of the microbes and prevent them from spreading further. But this is typically how you'll get a salmonella infection, is through the oral fecal route. Not typically because of human contamination. A lot of times it's because of animal contamination. Any questions about either of these articles? This one is a little bit more troubling. This is current event number 8, states citrus under threat. After ravaging Florida's crops for years, disease arrives in California. In Florida, it started with 2 dying trees in 2005. A couple of years and billions of lost dollars later, the killer, Huanglong bean, is in every citrus-producing region in Florida, is now in California. Huanglong bean, also known as HLB, is a virus. It's a virus that is transmitted. Let me continue this. This is a fatal disease, said Steve Lyle of California Department of Food and Agriculture. It's a very serious threat and one that we're taking extremely serious. The CDFA, the U.S. Department of Agriculture, and the citrus industry are teaming up to investigate the first HLB-infected citrus trees in California found in Los Angeles County. Sap-sucking insects called Asian citrus psyllids transmit HLB. These are little moth-looking organisms, brown and white. They are described at about the size of an ant. The bacterium, I said it was a virus, I'm sorry. It's a bacterium, ruins fruit and kills trees within a few years. California's citrus crop is worth $1.5 billion in 2010. And then there are a lot of us that have citrus trees in our backyards. So we're talking not about just oranges, but lemon trees as well, grapefruits as well, all susceptible.
I'm sorry, was it a bacteria or a virus?
It's a bacterium. I said virus, I misspoke. Sorry about that. That's what the article says. HLB bacteria. Yeah.
Is that a vector then, moth, or that mechanical?
Yeah, the moth would serve as a vector to transmit it to the fruit. Absolutely. Okay, so Asian citrus psyllids, which look like brown and white moths about the size of a red ant, live in subtropical regions across the world. They crossed into California about 2008 from Mexico, but this is the first time HLB has struck. The insects landed in Florida in 1998, and with hitchhiking HLB following in 2005. HLB didn't waste time. Just 6 months after agricultural agencies found 2 sick trees in Florida, it had moved into 12 counties less than 2 years later. It was in all 30 citrus-growing counties. In less than 2 years, it was in all 30 citrus-growing counties. So there's going to be a quarantine on the movement of fruit in California. Not that there already hasn't been. You can't bring food into California, but now you're going to see a quarantine on the movement of fruit between counties. So some counties will be shut down so that you won't be able to.
Just citrus fruit though?
That's what it targets, citrus fruit. So oranges, lemons. You might even see the price of citrus fruit start to go up. When I say might, you will see the price of citrus fruit go up. Any comments, any questions? Yeah.
What happens to the person?
Oh, nothing happens to us. We could eat the bacteria and it wouldn't affect us at all. You know, it just impacts the fruit trees.
Do you think they'll do that spraying like they did back in 2008 where they were spraying our area because of those false water?
Absolutely, they'll probably start. I don't know. You know, we don't want to jump the gun on what procedures they're going to take, but you know, the idea is you got to control the vector. You know, if this Asian moth is spreading the vector, you control it, you can control the spread of the disease. And so I'm sure that's the approach that they're going to take. And whether it leads to spraying, I don't know, probably. So last time we were talking about HIV. I'd like to continue our discussion. We'll start by actually looking at the structure of the molecule. Good. Looks like we'll have a video later today too. So, HIV, when we look at it, when we look at the microbe itself, the virus itself, what we see is that, of course, it's got an envelope that surrounds it. And this envelope, a phospholipid bilayer, is coming from where? Coming from the host cell, the cell that is kind of infected. In addition to that, we're going to find inside a capsid. We have a capsid, like viruses do, an envelope, and then within this capsid, you'll find 2 strands of RNA. In addition to that, there's going to be some proteins. One of these is reverse transcriptase. One of these is integrase. And the third is protease. These 3 enzymes are essential for the virus to be able to replicate itself. Essential in the infecting of the cell and in the replication of the virus. A couple of other things we want to note here. On the surface of the viral envelope here, we're going to find some surface proteins called glycoprotein 120 or GP120 and glycoprotein 41, or your book refers to it as transmembrane protein 41. Any questions about the structure here? So now, I should give you a chance to ask, right? So now, if we look at the cell that it's going to infect, the cells that are targeted by HIV are which ones? CD4 cells, which happen to be helper T cells. Macrophages also have CD4s. Some intestinal cells, some neurons also contain CD4 receptor sites. These are receptor sites on the surface of the host cell now. It is this receptor site that the virus is going to bind to. Now, there's not just the CD4 receptor. There's an additional co-receptor that we find on the surface as well. Now macrophages, this co-receptor is called CCR5, and for T helper cells, it's CXCR4. But the co-receptors both do the same thing. They aid in the binding of the virus to the surface of the host cell. They aid in the attachment. So now what happens here is when this virus, you've been infected with it, it'll come in contact with the surface receptors here and bind to the CD4 surface receptor with the aid of the co-receptors, and now the virus is bound to the host cell. A couple of things to keep in mind, or one thing mainly to keep in mind here. These co-receptors here are not there for HIV. They're not there for HIV to bind to. HIV has taken advantage of the fact that it has evolved molecules that will fit into these receptor sites. Okay? If we could change these receptor sites, we could prevent infection. In fact, my understanding is about 1% of the population lacks co-receptors, lacks the CCR5 co-receptor. Okay, so if you don't have